Welcome to today's lesson on protein synthesis and how changes in DNA structure can affect the protein synthesized by a gene. In our roadmap for today, we will begin by understanding the basic process of protein synthesis, including the roles of transcription and translation. Following this, we will delve into how mutations or changes in the DNA sequence can lead to alterations in the structure and function of the proteins produced. So far, you have learned about the structure of DNA and the concept of the genome. You have explored how DNA consists of nucleotides arranged in a double helix, and how the sequence of these nucleotides encodes genetic information. This knowledge allows us to introduce the new concept of how this genetic information is translated into functional proteins, and how any changes in the DNA sequence can lead to significant biological impacts. By understanding protein synthesis, we advance our comprehension of genetic expression and the molecular basis of traits, diseases, and evolutionary processes. Understanding the concepts of DNA in the genome helps us appreciate how traits are inherited, how genetic disorders occur, and how groundbreaking projects like the Human Genome Project have revolutionized medicine and biotechnology. Let's dive into the remarkable process of protein synthesis, which translates the genetic code in DNA into functional proteins. The first step in protein synthesis is called transcription, which takes place in the nucleus of the cell. DNA cannot leave the nucleus of the cell, so it has to be copied onto messenger RNA, mRNA, in a process known as transcription. Here's how it works. The DNA molecule unwinds and unzips at the base sequence of the gene that is being transcribed. Enzymes help in creating a complementary strand of messenger RNA from one of the DNA strands. Remember, in RNA, the base uracil replaces thymine. So A pairs with U, and C pairs with G. Once the mRNA is formed, it detaches from the DNA and exits the nucleus through the nuclear pores, heading towards the ribosomes in the cytoplasm. The process of translation occurs on structures in the cytoplasm called ribosomes. When the mRNA reaches the ribosomes, translation occurs. Here's a step-by-step breakdown. The ribosome reads the mRNA sequence in sets of 3 bases known as codons. Each codon specifies a particular amino acid. Transfer RNA molecules bring amino acids to the ribosome. Each tRNA molecule has an anticodon that is complementary to the mRNA codon. This ensures the correct amino acid is added to the growing protein chain. The ribosome moves along the mRNA, reading codons and adding corresponding amino acids brought by tRNA. This process continues until the ribosome reaches a stop codon, signaling the end of protein synthesis. After translation, the newly formed amino acid chain, also known as a polypeptide, folds into a specific 3-dimensional shape. This unique shape is crucial because it determines the protein's function. For example, enzymes are proteins that act as biological catalysts, speeding up chemical reactions. Another example is maltase, which breaks down maltose. Enzymes requires a specific shape, so that their active site fits specifically with its substrate. Hormones are proteins that carry signals throughout the body, like insulin, which regulates blood glucose levels. They require a specific shape to be recognized by cell surface receptors on their target organs. Structural proteins provide support and strength, such as collagen in connective tissues. These also require specific structure for their function. The DNA sequence is critically important for proper protein synthesis. But what happens if there's a change or mutation in this sequence? Insertions are a type of mutation where an extra base is inserted into the DNA sequence. This causes a shift in the reading frame shift mutation, potentially altering every amino acid downstream. This means that the codons are now read one base along from where it originally was, meaning that different amino acids might be joined to the polypeptide sequence by tRNA. Deletions mean a base is removed from the DNA sequence, also causing a frame shift and leading to widespread changes in the amino acid sequence, and, consequently, the protein. A substitution mutation is when one base is swapped for another. This might only change one amino acid and does not usually affect the reading frame, but can still alter protein function. While many mutations are neutral or minor, some can have significant effects, particularly if they occur in important areas of the gene. Some mutations result in a protein with an altered shape. This can affect how well the protein functions. For example, an enzyme with an altered active site might not bind properly to its substrate, reducing its efficiency or rendering it inactive. In some cases, the protein might not function at all due to the mutation, which can lead to disease or dysfunction. Understanding protein synthesis and the impact of mutations provides vital insight into the functioning of our genes and the development of genetic disorders. This knowledge is foundational in fields such as genetics, molecular biology, and medicine. Let's work through a question to understand protein synthesis. In this question, we need to describe the basic steps of protein synthesis. We start with transcription. In the nucleus, a segment of DNA is used as a template to produce mRNA. RNA polymerase binds to DNA and assembles a complementary strand of mRNA. The mRNA strand leaves the nucleus and moves to the ribosome in the cytoplasm. Next, focus on translation. At the ribosome, the mRNA sequence is read in codons, 3 nucleotide sequences. tRNA molecules bring amino acids to the ribosome, matching their anticodons with the mRNA codons. Amino acids are linked together to form a polypeptide chain, which folds into a functional protein. Therefore, protein synthesis involves transcribing DNA to mRNA and translating mRNA to a protein. In our next question, a section of DNA had the base sequence A, T, C, G, A. What would be base sequence of the section of mRNA that has been transcribed from this DNA? First, it is important to remember that the complementary base to C is G, and T is A. In mRNA, the base uracil is used instead of thymine, so A is complementary to U. Therefore, the complementary mRNA sequence to the DNA section would be U, A, G, C, U. In today's lesson, we covered protein synthesis and how DNA changes can impact protein production. We discussed the 2 main stages: transcription, where DNA is used to create mRNA in the nucleus, and translation, where the mRNA is read by ribosomes to assemble amino acids into proteins with the help of tRNA. We also examined how mutations in DNA can alter the protein produced by a gene. Even small changes in the DNA sequence can affect protein structure and function, leading to varying impacts on the organism. This understanding of protein synthesis and mutations provides insight into genetic variation, trait diversity, and potential genetic disorders.